In our BioTuring ecosystem, we provide 2 versions of Lens for spatial omics analysis. Choose the suitable platform for your bulk spatial data or single-cell spatial data. You can choose between your personal workspace or shared workspace between you and your colleagues. Before upload your dataset, make sure to select the right species for your data: human, mouse, or non-human primate. Other cases, please choose others. To add your in-house data, click on Add Studies. In Lens for bulk spatial, we support nanostring geomics data, 10x Space Ranger output, Syrah object, Scampi object, and Curio data. In Lens for single-cell spatial, we support both spatial transcriptomes such as technologies like Cosmix, Xenium, Merscope, etc., or spatial proteomes such as Codex, SciSif, MIHC, and other spatial technologies that have expression quantification with images. Select the file format of your choice. The study info is optional. You will have a quick guidance for you to select the suitable files. Additionally, you can include multiple batches simultaneously. You have different ways to upload your data, either from your local computer, by a shared directory between you and your colleagues, or via Amazon S3 platform. Select all the files according to the guidance for the format you choose. In Lens for single-cell spatial omics, you also have the option to either use the default filtering parameters or use your own. For more detail, you can view our instruction here. You can rename your batch here. Agree to upload your data to BioTuring Cloud and click Submit. After submitting, your studies will be stored in this My Studies section. You also can share the study with your colleague by click on this button. The studies shared by your colleague will be stored in this section. Beside, the public studies searched and saved from Talk 2 data will be saved here for you to quickly access. We hope that you find this video helpful. For more information, please contact us via support@bioturing.com.